most of you. So I'm an associate professor in cognitive science. And actually, I feel sort of funny, talking as a bioinformatician. So my training, actually, I studied physics as an undergrad and did my PhD in neuroscience. So I really consider myself a computational neuroscientist. I was telling Aditya that I did a postdoc at the Salk Institute before coming to UCSD in Terry Sanowski's lab, working on analysis of EEG data. But when I was there, when I was at Salk, I started collaborating with some folks studying the epigenome of brain cells, and I'll tell you about that work today. And so I sort of came into bioinformatics from that direction. And I still consider, so at this point, I suppose I am a bioinformatician. I do, most of the research in my lab is bioinformatics related. But I think of myself as being defined by the questions and the problems we're trying to address more than the methods. And so that, we're really neuroscientists. We're interested in how the brain works, how cell types and circuits function in the brain. And we happen to be using some, genome sequencing, DNA sequencing, RNA sequencing data to answer some questions. But we're really, so I kind of define myself more in terms of the neurobiology hat, but I guess I wear both of those hats these days. So I know this is supposed to be a chalk talk, and so I tried to say, well, let's see, I pasted some papers to my wall, and I might try to use that as a kind of whiteboard later, but maybe I'll just start out with some slides. So let's see if I can share my desktop. So you guys can see my screen?
Yeah, we can see that.
Here we go. Okay, so I'll give, I don't want this to be an all PowerPoint kind of presentation. So please interrupt me at any point if anything is unclear or you have questions, and I'm happy to chat and discuss. Let's see, was there something in the chat? Okay, great. Awesome. So, and actually what I'm going to do, I'm just going to share this screen. Okay. So I'll talk today about integrating single-cell transcriptomes and epigenomes for the purposes of characterizing brain cell type diversity. So this is a line of work that my lab has gotten very involved in over the last few years, and that several of my lab members are involved in. Let's see, if I go to full screen, can you still see my slides now?
Yeah.
Yes.
Yeah. Okay, do you see a blank slide now?
It hasn't moved. The slide hasn't moved.
Yeah, that's what I thought. Yeah, no, it doesn't look. I have to do it this way. Let's see. I have to share the full desktop. Okay. Okay, cool. So my lab has a number of areas that we work in. So just the big picture overview of my lab is we're interested in understanding the identity of different brain cell types by looking at their epigenomes. We're also interested in, at the other end of the spectrum, we're interested in plasticity. So how, in particular, the epigenome contributes to brain cells changing over time. So we know that changes in brain cells, changes in connections between brain cells and in the cells themselves are important for learning, memory, adaptation, and aging. We're interested in epigenetic diversity, by which I mean, what makes two siblings different from each other? They're similar but different. They share a lot of genetic commonalities, but they also are distinct individuals. And we think that in part is due to epigenetic differences in the brain. And overall, we're interested in how can we leverage computational and data science methods to really address these questions at the scale of the really complex brain circuits that are present, especially in human brains, mammalian brains, where you have 100 billion neurons. One common thread through all of our research is our interest in neuronal DNA methylation. So I'll tell you about this. Basically, DNA methylation is one of the best studied epigenetic marks, and we've really been able to take advantage of it to address all of these areas. So you know what? I'm gonna skip some of this intro stuff just in the interest of time. Happy to talk more about it. Maybe I'll start here. So what are cell types? Well, I mean, I guess you guys all know that there are differences between skin cells and lung cells and muscle cells. Traditionally, if you open a textbook that it talks about cell types, traditionally a neuron might be considered one type of cell, right? It's a very important type of cell. Most of the, the information processing in the brain happens in neurons. But it turns out we now appreciate that there's not just a single type of neuron. In fact, there are hundreds and probably thousands of different types of neurons. And these types have lots of different properties. So one, striking difference is their different morphology. So here on the top, you can see tracings of the dendrites and axons. So the light gray colors are the dendrites of a single cell, and the dark gray are axons. Or actually, I think it's the opposite. I think that the light gray are the axons. But basically, the input and output tree-like structures. And these aren't just interesting patterns. The fact that they have different shapes has very important consequences for their function. And I won't get into that now, but that's one sign that these are very different. You can find differences in their electrical properties. So this is electrical recordings of these different types of neurons. And you can see when you inject a current, a small step of current, you get a different type of response in each of these different types of cells. And most importantly, for our purposes, they're very different at the molecular level as well. So they all, even though they all have the same, genome, they all share the same DNA in their nucleus. The pattern of gene expression in each cell is different. So SNAP25, for example, is a gene that's expressed in all neuron types. And so you can see, so these are called Violin plots. Maybe you've come across these. Basically, the height of each of these plots shows you the level of gene expression in a different cell type. And each column is a different cell type. So what you can see is that they all express SNAP25, only some of them express this gene GAD1. Well, this is marking inhibitory cells. So by looking at these patterns of gene expression, you can start to disentangle different types of neurons. And this is just an example from a couple of years ago where people looked at retinal bipolar cells. Anybody know what a bipolar cell is in the retina, by chance? So you guys may know that the retina, the retina, of course, is the part of the brain that happens to be on the back of your eye. So, but it's considered a part of the central nervous system. Yeah, these are neurons. That's right. They're they don't have two axons, but well, I guess you might say that. I mean, actually, they don't have axons at all. They just have dendrites. So that's a kind of funny thing about these bipolar cells. But the retina, the way the retina is constructed, it has photoreceptor cells, which are the ones that actually absorb light and turn that into electrical signals. Those then synapse onto some interneurons that pass the information finally to the output cells of the retina. And so the bipolar cells are a kind of interneuron. They're kind of intermediate messenger within the retina. And you can see this is a plot, and I'll talk more about what this is showing, but basically you can think of these clusters as different molecular cell types. Each dot here is one cell, and they're clustered by, their similar patterns of gene expression. And you can see there's really dozens of different types of bipolar cells in the mouse retina in this case. If we zoom out to the level of the whole nervous system, this is a study from a few years ago where they did single cell RNA sequencing. So how many of you guys has anybody heard of single cell RNA sequencing? Have you had a talk about it in the past? Possibly. So, so you probably know that in single cell RNA sequencing, you're capturing the transcripts and sequencing them to determine what genes are being expressed. So in this case, they did that for 500,000 individual cells across different parts of the mouse central nervous system and peripheral nervous system, actually. And they came up with something like 250 different cell types. So one of the questions that we're interested in is how do we take advantage of this really large scale, these types of large scale data? Those studies I just showed you are from a few years ago. We now routinely work with data sets that have, millions of cells, and not just large numbers of cells. They also can have lots of different types of information. So I'll show you some of I just showed you RNA sequencing information. We also have epigenetic information, and we want to, combine all those together into an understanding of cell types. But the question arises, are we done yet? So have you guys heard the term lumpers and splitters? I actually have a cartoon of this on the next slide. So the term lumpers and splitters goes back to Darwin, actually. So Darwin said it is good to have hair splitters and lumpers. And, I think he was thinking of, species, and he would go out and classify the species of beetles that he found, or of zebra finches. And when you're doing that, depending on your mood or your personality, you might choose to group things into large classes where these are all just birds, or you might feel more like a splitter in which you say, okay, well, every one of these particular distinctions with a slightly different beak shape or beak length is a distinct category, or maybe even a species. And there's another quote that I like from 100 years later from George Simpson. He says, splitters make very small units. They can tell two animals apart, but if they can tell two animals apart, they place them in different genera, and if they cannot tell them apart, they place them in different species, right? So he's kind of mocking people who are overly splitting. Lumpers make larger units. If a carnivore is neither a dog nor a cat, neither a dog nor a bear, they call it a cat. So, those are extremes, but, the question arises when we're doing this single cell work, at what point do we stop splitting? And when are we done? So one of the questions in my lab is how do we come up with computational methods to try to address that question, to do some kind of cross-validation to tell us whether a split of a category is overfitting or whether it's appropriate to the data. Okay, so let me see what what I wanted to tell you about today is our work using single-cell DNA methylation. So I'll start by sort of describing single-cell DNA methylation data and show you some of the stuff that we've done with it. And then I'll describe, since this is a bioinformatics group, I'll try to describe for you a method that we've developed for integrating different data sets. But again, my goal is not necessarily to get through everything I had planned. So please stop me if anything is unclear. I'm very happy to spend the time discussing and explaining. So the reason, as I think you guys probably have appreciated already, the reason for looking at single cells is because when you take a sample of tissue, you're looking at a smoothie, right? So, and this is what was done throughout most of the history of, studies of cell types using molecular methods. Simply because it was impossible to capture the DNA or the RNA, the genetic material from a single cell, and then efficiently amplify it in such a way that you could sequence it. And when you do that, you're losing the identity of all of the different cells. So for example, there could be a gene that's expressed equally in all of the cells, and another gene that's expressed strongly in one cell type and much less in another cell type, and those would average out to give you the same signal. So a few years ago, our collaborators, Joe Ecker and Marga Behrens at the Salk Institute, and my student Chris and our collaborator Chun-Wan, we all collaborated on the development of this method for doing something like single-cell RNA sequencing, which I've just shown you and which you guys are probably familiar with, and being able to do it for sequencing DNA methylation. And the basic idea is you start with tissue. This is kind of some details. You actually isolate the nuclei of the cells, and it turns out it's a lot easier to isolate nuclei from the brain than it is to get whole cells. And that's an advantage for DNA methylation because we only need the nucleus. We don't need to capture all the RNA that's in the dendrites. We actually then sort individual nuclei into single wells of a 384 well plate, shown here, and then do some tricks during the library preparation to tag every cell with a unique barcode, followed by amplification and sequencing. Now, the one key step for being able to read out DNA methylation is called Bisulfite Conversion. Have any of you come across Bisulfite sequencing before? So if you haven't encountered bisulfite sequencing, the idea is that you treat the DNA with sodium bisulfite, and what that actually does is it converts cytosines into uracils. And so when you do the sequencing, then wherever there was a C in the sequence, you sequence it as a T actually. But the trick is that methyl cytosine, if the cytosine had a methyl group on it, that's protected from this conversion. So those methyl cytosines don't get converted. So you sequence those as Cs, and therefore by looking at the sequence data that you get out, you can infer which sites were cytosines and which were methyl cytosines. Does that make sense? Any questions about that? I'm kind of going over that quickly, but stop me if you have any questions. So once we have the data, we want to use it to visualize kind of the pattern of gene expression. And this is something that's similar to, the pattern of cell types. And this is something that is similar to what's done in single cell RNA sequencing. We have essentially a dimensionality reduction problem. So let me see if I have here a picture of so you can think of the raw data that we get out as a matrix of several thousand bins. You can think of something on the order of 30,000 bins by several thousand columns, and each column is a single cell. So for each column, we have the methylation level, the amount of DNA methylation in a certain region of the genome. And our hypothesis is that different cell types are going to have different patterns. So maybe cell A and cell C are both of the same type, and so they have a similar pattern of DNA methylation, but cell B is a different type, and so it's going to have a different pattern. The problem is that this data set lives in a high dimensional space, in about a 30,000 dimensional space. So we don't have any way of kind of visualizing what this pattern is. I can't show you a 30,000 dimensional plot. And so we use these techniques for dimensionality reduction. So I'm sure you guys have come across TSNE and UMAP, for example. There's other methods, variational autoencoders. There's now a whole industry of folks that are interested in methods for embedding data from high dimensions into lower dimensions. I always like to illustrate the concept with this idea of taking the globe. We know that the Earth is a three-dimensional, is a flat. Is the surface of a sphere, right? So it's a two-dimensional manifold that's embedded in 3 dimensions, right? So to really accurately represent the whole globe, you would need a globe. You'd need a 3-dimensional embedding. However, it's very convenient to make 2-dimensional maps like these. These happen to be maps of the whole globe, but of course there are compromises. There's no way to do this without tearing the edges of the world. So here, for example, in the middle of the Pacific Ocean, we have something that's very, very inaccurate. So this particular projection of the globe is pretty accurate here around the equator. And it also happens to be, it's trying to preserve kind of distances as much as possible around in the center here, but it gets wildly inaccurate here in the middle of the Pacific. Here's a different projection that makes a different trade-off, but it still is going to have global inaccuracies. So I think that's an important thing to keep in mind with these projections. And so we can do that and apply that to our data. Many of you have probably come across principal component analysis. That's a way of projecting data. It happens to be a linear projection method. So if you think about it, if you had data that lived, so imagine that each of your single cells was a point on the surface of a 3-dimensional, on the surface of a globe. If you tried to do PCA, which looks for a flat plane to project it onto, there's no way, there's no single flat plane that can capture the whole globe. You would end up squashing the North Pole, so it's sitting right on top of the South Pole, for example. So PCA applied to our DNA methylation data ends up looking like this, where we can see some cell type distinctions, but there's a lot of groups of cells that are kind of mixed with each other. If we use something like TSNE, and again, feel free to ask me more about what exactly is going on with TSNE or UMAP. We can talk about that later. But for now, I just want to say that what this is able to do is to more clearly separate out groups of cells that are more similar to each other. And it does it by finding a curved manifold, a curved surface in the high dimensional space. Okay, so I'm going to skip this because what I want to tell you about what we then can do. So, there's more that we could talk about. We can use these data to look at individual genes, to look at individual groups of cells to try to figure out what they are, how are they connected to, the classical cell types that we know about in neuroscience. But what I wanted to focus on is an effort that we've been making to try to combine data from different single cell modalities. So what I just showed you are single nucleus methylcytosine sequencing data. So this is the DNA methylation data here in blue. We have data in this case from about 10,000 cells, all in the motor cortex of the mouse. So this is a picture of a mouse's brain. For those of you that aren't familiar, here's the front of the mouse. This is a region called the olfactory bulb. You can see the brainstem back here. So this strip over here is the mouse motor cortex. Again, we've sequenced about 10,000 cells for DNA methylation, and for each cell we get about 1 million or 1 or 2 million individual fragments, reads, sequences of DNA. And that it works out that covers about something like 5 to 10% of the genome for each cell. We're doing this particular data set was collected in the context of a big collaboration with several other groups, actually around the whole country, that are also focusing on motor cortex, and they collected other kinds of data. So for example, some of the groups collected single cell RNA-seq using a technology called SMART sequencing. Don't worry too much about these acronyms. This is just a particular platform for doing single cell RNA sequencing. And single cell RNA sequencing, again, captures a few thousand cells. The number of cells you can capture is basically limited by the cost. So the cost here is around, on the order of dollars per cell. So that's why you can't do like 1 million cells. But you get millions of reads per cell. On the other hand, there are other technologies like something called 10x. Some of you might have heard of 10x sequencing. So 10x is a company that makes very high throughput technology for RNA sequencing, where you can do, in this case, 100,000 or up to 150,000 individual cells, again, from the motor cortex of the mouse. But for each cell, you only get, in this case, 10 to the fourth or 10,000 individual molecules that you're measuring. So there's this kind of complementarity, and in addition, we're measuring different things. Here I'm getting DNA methylation, here I'm getting gene expression. Here's another technology called ATAC-seq, some of you may have heard of. Has anybody heard of ATAC-seq? Anybody want to try to explain what ATAC-seq does? I'm sure there's somebody out there that's heard of ATAC-seq. And don't worry, this isn't for a grade, so. Okay, that's honest, Crichton. I appreciate it. So, so ATAC-seq is measuring what we call accessible chromatin, right? So Aditya says sequencing a specific sequence. Oh, that's a good guess, but not actually. So ATAC is an acronym. It stands for assay of wait a minute, assay of transcriptase accessible chromatin. So what you're doing is you're using a transcriptase called TN5 to find regions of DNA that are accessible or open. And those mark kind of active promoters and also enhancers. So it's another way of looking at the epigenome. Let's not worry about it too much for now, but just to say that that's another epigenomic measurement. And again, it has this, it falls along this spectrum of high throughput versus, high coverage versus high throughput. In this case, ATAC-seq is more high throughput in terms of getting a large number of cells, but a fairly limited number of measurements per cell. So the challenge that we had in the context of this project was to try to take these different types of data and combine them. So if we look at just the RNA sequencing data, here you can see an embedding. This is like the t-SNE that I showed you earlier. In this case, we're using something called UMAP, which is a similar idea. Each dot is a cell. We're coloring them by which cell type we think they are. Some of them are astrocytes, oligodendrocytes, and then these are all different kinds of neurons. so we get this kind of picture and we see what cell types there are. But we can do the same thing with the DNA methylation data or with the open chromatin data from ATAC-seq, and each of them gives us its own map. But of course, these are all different maps of the same landscape. So, it's like I have a satellite map of California, and I also have like a hand-drawn map or a map that's made by, in some other way. What I really want to do is combine all of those different views into a unified representation where I know, for example, I have this whole spectrum of, these happen to be excitatory neurons that are in different layers of the cortex. I want to know how the DNA methylation patterns for these neurons match up with the transcriptome, with the RNA sequencing patterns. So how can we do that? Well, so the way we think about it is like this, and this is where I really wish I was able to use an actual chalkboard. I would draw it out for you guys, but so the way you can think about the data set is, I showed you a matrix for the DNA methylation data, right? So let's look at DNA methylation. That's single nucleus methyl C-seq here in green. So every row is going to be a cell, and every column is a feature. In this case, the features might be genes, or they might be bins throughout the genome. And so with the DNA methylation data, I have some cells, and I've measured these, I've made these measurements. Now, of course, these cells have a transcriptome. They have a gene expression pattern, and they also have a pattern of chromatin accessibility. It's just that I don't know what those patterns are, right? So this question mark indicates that it's missing data. Basically, I have some data that I'm observing, but there's other data that are out there, but I'm just missing. And unfortunately, it's very difficult to make the measurements simultaneously in the same cell, although there is progress on that front, and there are ways of maybe measuring, both DNA methylation and gene expression in the same cells. But for the data set I'm showing you here, these are measurements that are made separately in different groups of cells. So similarly with the transcriptome data, I have a group of cells for which I know the transcriptome, I know the gene expression, but I don't have any information about the corresponding DNA methylation or chromatin accessibility. And finally, I have something for the ATAC-seq side on a certain group of cells, but I'm missing the corresponding information about the transcriptome and the DNA methylome. So the trick that we're going to use to try to integrate these data is to take advantage of the fact that they're correlated with each other. And so you can kind of see that here. So this is a particular gene called TAC1. Now, TAC1 is expressed strongly in one particular type of neuron called P-valve relin calb1. Don't worry about the name. It's a particular type of inhibitory neuron. So you can see the SMART-seq RNA sequencing data here has lots of reads. It also has a high level of chromatin accessibility. So this is the ATAC-seq in blue, and it has a low level of DNA methylation. And that makes sense because we know that DNA methylation is a repressive mark. So whenever we see very low methylation in the gene body, that tends to correlate with having a high level of gene expression. So the idea that we came up with, and so this is work from Feng Ming, who I think might be on the call in my lab, together with Ethan and former postdoc Wayne Doyle. We came up with a method for taking advantage of these correlations to build a graph to link the different data sets. So imagine that you have, your methylation data up here and some RNA sequencing data down here. Again, these are different cells. What I'm drawing here is a two-dimensional representation, so you can think of this as like a T-SNE or a UMAP. But these are different two-dimensional representations. They're different maps, and so I don't know how these coordinates relate to these coordinates. So what I can do is I can look for each cell, for each of these green cells, I can look for which are the purple cells. None of the purple cells is going to look exactly like it because I'm measuring the DNA methylation. I'm not measuring the gene expression. But I can find the ones whose DNA methylation is most strongly correlated with this green cell. And so I can build a graph of nearest neighbors. So this is sometimes called a k-nearest neighbors graph. And I can also go in the other direction. I can ask for each purple cell which green cells have the most correlated pattern. Now there's a few subtleties that come in here. If you've ever worked with this kind of k-nearest neighbors graph, you have to you have to be careful that you don't end up kind of overfitting the data in a way where, for example, if there's one cell in this green data set that has higher, more noise than other cells, maybe it's got a particular noisy measurement. In some cases, that can end up being a hub cell where all of these purple cells end up looking most similar to this one green cell. So we have to play some tricks to avoid those kinds of things. For example, by looking for cells that are mutual nearest neighbors of each other, or using other tricks. But once we do that, what we can do is, so Aditya is asking, is this a machine learning approach? Yeah, I would say it's a kind of machine learning. I would say it's more like traditional machine learning and not deep learning. So this doesn't involve a neural network. And it's sort of unsupervised. So what we're doing is we're just building a neighbor graph and using that to link these two data sets. So, and it's non-parametric. So, yeah, it's an example of, I guess, unsupervised manifold fusion, manifold integration. So once you have this graph built, what you can do is you can impute the missing data. So for example, for each of these cells, we want to know what would its methylation pattern have been like if I had been able to measure it. And so what we do is we just take, for example, this cell, we take its nearest neighbors over here and we average their methylation pattern to infer what the methylation pattern would have been here. And we can do that in the other direction as well. And so once we've done that, now all of these cells have compatible data, right? I have a full data set, both RNA and DNA methylation for these cells and for these cells. And so we can go ahead and now do joint embedding and clustering in the same space. And so, just to show you that it kind of works, this is Feng Ming went ahead and applied this to 8 different data sets. So there's actually 6 different RNA sequencing data sets plus ATAC-seq and DNA methylation. And here, and it's over, I think, 600,000 cells. And what you can see is each dot here is one cell and they're colored by which data set they came from. And so what I think you can appreciate is that the colors are all nicely mixed together. They're not separating by which data set they came from. If we didn't do this whole nearest neighbors imputation thing, if we just, for example, took all of the data sets and tried to run t-SNE on them, for example, just treating the gene, without doing any correction for the fact that they come from different data sets, what would have happened is that they would have completely separated. You'd have data set 1 over here, data set 2 over here, data set 3, etc. But instead what we're getting is we're getting separation by cell type. So you can see if I now recolor them by which cell type they come from, you can see that for example, these SST neurons are all orange over here, but it includes cells that came from each of the different data sets. And then we can use those data to go back and take, for example, I can take this group of cells, this cell type, take all of the methylation samples that contribute to this cell type and put them together into what we call a pseudo bulk sample. So put them together into, just combine all few hundred of those cells so that we now have a very high coverage estimate of the DNA methylation pattern for that cell type. And we can go ahead and analyze what is the pattern of DNA methylation for that cell type across the whole genome. So let me pause there. I've thrown a lot of stuff at you guys. Well, I guess I can go through this animation real quick. So this is showing a genome browser that shows you in green the DNA methylation patterns. Each row is one cell type. So each track is one cell type. And then in blue here we have ATAC-seq, so open chromatin again for the same cell types. We can zoom in on them. And this is, again, that locus that I was showing you earlier with TAC1. So here's the TAC1 gene. You see that this one cell type has low DNA methylation, high chromatin accessibility, and high gene expression, whereas you have these other cell types that have much higher DNA methylation, lower accessibility, and no gene expression at all. one of the really cool things about this data set is that now, not only can we map the cells into the same space, that's all well and good, but so what? What can I do with the data? Well, one of the things that we're really interested in doing with it is using it to study not just which genes are expressed, but how they're being expressed, right? What causes this gene to be expressed in this cell type? Well, one of the main models we have for how a cell type specific gene expression is controlled is that there are these distal regulatory elements. So sequences that are far away from the actual gene or the gene promoter itself. So here's the promoter of the TAC1 gene is over here. This location is about 20,000 base pairs upstream of the gene promoter. And what you can see, so this is, there's no gene in this region. This is an intergenic region. There's no gene bodies nearby. But it has an attack-seek peak. And so what that tells us is that this is an accessible region. It's a region where the nucleosomes, the histones have been pushed aside, presumably to make way for transcription factors to be able to bind there. And so we know that these kind of accessible regions, and in addition, it's a little bit harder to see on this browser, but there's a low level of DNA methylation at this location. In particular, it's lower in this cell type than it is in the other cell types. And so to us, this is a signature of an enhancer that's cell type specific. It's a regulatory element that's active specifically in this cell type and not in the other cell types. And you can see that that correlates very well with the expression of this gene in the very same cell type. So the hypothesis here is that this is an enhancer that is controlling this gene. Now, how could that happen? What do you guys think? How can an enhancer that's 20,000 base pairs upstream actually control the expression level of this gene? How do you think the message gets from here to here?
It could promote chromatin folding or compaction.
Great, exactly. Yeah, who was it that said that?
Me, Bhupala.
Okay,
I'm
great.
the one with the long name. Let me change that later.
Okay. Did you say Bhupala? Is that how you say it?
Yeah, Bhupala.
Bhupala, great. Yeah, that's exactly right. So there's so the DNA inside the nucleus is not stretched out. It's of course folded in 3 dimensions. And so what happens is that you can get loops of chromatin that are formed that might bring together this particular regulatory region with the promoter of this gene. We don't have data about that happening in this particular cell type, but there are other types. So you guys might have heard about techniques like Hi-C or 3C that are ways of actually trying to measure the chromatin conformation directly. And so our hypothesis would be that if you were able to do that experiment in this particular cell type, you would find a loop connecting this enhancer with this promoter. Okay, now I want to pause there before I could go into this next topic about cross-validation, but let me pause there. Are there any questions about kind of data integration? And so yeah, so is it Jan or Jan? So first one, Jan. Okay, so Jan is asking, is this a tool for discovering enhancers and silencers? Yeah, exactly. So that's really what the primary motivation for collecting the DNA methylation and attack-seek data is so that we can discover these enhancers. And does anybody have any thoughts on why should we care about discovering cell type specific enhancers in the brain, for example?
Might be a drug target, for example, if
I think what makes the gene expressions different is those enhancers, right?
Okay, so I'm getting two. Aditya and Ishan. So Aditya said that it could be a drug target. So on a practical, I really like how you're thinking that on a practical level, if we want, for example, to correct for problems in the expression of a particular gene, it might be useful to know which enhancers are regulating that gene. Let me sort of take that one step further and also talk about genetic contributions to disease, right? So we know that, you guys are familiar with GWAS data, right? Where people look at which genetic variants are associated statistically with a particular disease. It turns out that most of those genetic variants in GWAS are not in coding regions. They're not in, they may not even be in a gene body. They may be, in an intergenic region that's far away from a gene. And what this kind of data can let us say is if, for example, if there's a genetic variant that overlaps this enhancer, and that is linked to schizophrenia, for example, or autism, that would then give us a hypothesis that maybe the reason that genetic variant is having an effect in that disease is because that enhancer is active in this particular cell type in the brain. And so it starts to give us a kind of a place to look for the pathway that's being directly affected by that genetic variant. Now, Ishan mentioned, I think you said something about that this is the way that the gene expression is regulated, and so we kind of want to understand that at a basic level. Is that what you said?
Yeah, in different cell types, we have different expressions based on those enhancers.
Yeah, exactly. So yeah, that's another really good reason for wanting to understand this. There's another one that has to do with creating tools for neuroscientists. So one of the things that neuroscientists really want to be able to do is to be able to study the brain by manipulating and recording from specific cell types. So traditionally, if you think about neuroscience, people might put an electrode in the brain or put an EEG electrode on the top of the scalp and record the electrical activity of neurons. But the problem with that is that you have no idea what type of neurons you're looking at. And as I've shown you, there are hundreds of different cell types in the brain. And you can imagine it might make a big difference. So for example, there might be one particular cell type that may represent a very small fraction of the total population. And we know that there are cases like this. We know that there are, for example, particular populations of neurons that are really important for regulating sleep, for example, or for regulating reward. And if you just go blind with an electrode and try to record from the first neuron you find, you might never stop to study that neuron. You might get, if it's 1% of the neurons, then you're very unlikely to ever realize that you're even recording from it. So what do you think? Does anybody, okay, so now that I said that, how do you think you might be able to take advantage of this knowledge of enhancers to actually target those cell types? Any thoughts?
You could knock out maybe the genes that express that enhancer and see how it affects the upstream.
Okay, so that's an idea. So you could, so for example, this is telling me that TAC1, this TAC1 gene is specifically expressed in this cell type. If I knock it out, maybe I can predict that that's going to have an effect in this cell type. That would really be, primarily that would be a way of testing the effect of this gene. But what if I want to not actually perturb the neurons, perturb the brain? I want to have a normal mouse, but I just want to be able to record from these neurons.
I mean, you'll have some kind of product that you can track the level of.
A product like what? What would be a really convenient product?
I mean, whatever the gene expresses, right?
Oh, I see.
But you'd have to know that as well. So
right. So I could try to go in and look for the genes that express TAC1. So maybe with an antibody or something, right? And use that to mark those cells. But what's a really, really convenient way of marking cells in tissue? So that could work, but that's difficult because then for every gene you have to find an antibody for it, and it's kind of messy. So what about making the cells glow?
Yeah, I think someone in the chat mentioned.
Oh, okay. Oh, yeah. Jan and Creighton. So yeah, so I want fluorescence. I want to make the cells light up so that I can look in my microscope and see which ones are glowing. And so how do we do that? Yeah, GFP or RFP. Yeah, I want to get them to express a fluorescent protein like GFP, right? So we'll get to optogenetics in a second, Jen. So hold that thought. So let's say I want to get these cells to express GFP. Now, I know how to get GFP. I can use a virus or a transgenic mouse to express GFP in cells. But what I want to do is express GFP specifically or only in these cells. So how can I do that? Again, taking advantage of this knowledge that I've just found that this is an enhancer that's active specifically in this cell. So basically the idea is I want to take advantage of the fact that the cell is already doing it for me. The cell has figured out a way of expressing this gene only in this cell type. Sorry, the body has figured out a way of suppressing this gene in all the cell types except for this cell type. So what I want to do is I kind of want to stick in a transgenic gene, stick in a GFP, either with a virus or with a transgenic mouse, but put it downstream of this enhancer or this promoter, right? And then the cell will take care of the rest for me because the cell already knows how to, if it's this type of cell, it knows that whenever it sees this enhancer, it's going to make it accessible and it's going to drive the expression of the linked gene. Now that, that all sound, does that make sense? Do you guys follow the logic? So, so that way I can get the cell to glow green. Somebody else mentioned calcium imaging. So that's a great idea. I could, instead of putting in GFP, I could put in, GCAM, which you guys might know is a genetically encoded calcium sensor. So it's a different kind of fluorophore whose fluorescence, it binds calcium. And when calcium comes into the cell, I'm able to see increases and decreases in the brightness that tell me about the activity of those cells. So, having an enhancer, a cell type specific enhancer, is the starting point for a lot of really, really important types of studies that you can do. So Jen, you mentioned optogenetics. How would this allow me to take advantage of optogenetics, and what would that give me over and above just GFP expression? Or anybody? Anybody familiar with optogenetics, with channel rhodopsin? Have you guys heard of? Yeah, so you can choose to activate the gene when you want to. What I would say is you can choose to activate the cell. So the way that optogenetics works is you can express a gene. So instead of expressing GFP, you would express a gene like channel rhodopsin, which is a light-gated ion channel. So what you do is, and you express it using this enhancer to drive the expression so that it's going to be expressed only in these cells. Then what you can do is just shine light on the brain. Sometimes you can even do it through the skull. You can just shine a really strong LED through the skull. If it's a deep part of the brain, you might have to put an optical fiber down. Shine light, and whenever you shine the light, it's going to activate those channels in the membrane of the cells that are expressing the channel rhodopsin. They'll open and depolarize the cell or hyperpolarize the cell and cause it to become active. So that can be a really powerful tool as well, because that way you can not only control which cells are being activated, you can control exactly when you're activating them, because you can control when you shine the light. And you can control that with millisecond precision. Okay, great. So what do you guys think? I don't know, what's the timing here, Aditya? Do you guys keep going till 6 or?
Yeah, so we have our GBM at 6, so I think we could do like a Q&A session for about like 10 minutes.
Great, yeah.
After that. So if anyone has any questions, just like raise your hand.
And let me just, maybe while we're doing that, maybe I'll just flash up my slide of my lab just to thank the lab members. So this is, these are my lab members, and the work I told you about was really, actually all of these people participated. Jun Hao, Christina and Wayne have moved on, but Feng Ming, Ethan, are still actively involved, and we collaborate with others. But yeah, please go ahead and any questions. Are any of you working in a lab? Aditya was telling me he's working in Terry Sinowski's lab. Do any of you what's the next step? Good question. So Jan, what do you mean by the next step? You mean the next step for our research, for in our studies? So one of the next steps, sort of linking with what I was just talking about, is we want to validate these. So this data set tells us that this enhancer, seems to be strongly expressed in this cell type, but it doesn't guarantee that what I just described is going to work. In other words, if we make a virus that contains this enhancer sequence, we want to check whether that means that that virus is going to drive expression specifically in this cell type. So we're working on a project with some collaborators who are actually doing some very clever assays to test these viruses, to sort of create viruses based on sequences that we've predicted from these data sets, inject them in a whole bunch of neurons, and then look at which neurons actually end up expressing which viruses. Using barcodes, you can kind of, in a multiplex fashion, test lots and lots of viruses at the same time, lots and lots of enhancers, and then look at the data that comes back and try to infer something about which enhancers are actually effective. So it turns out that, these data can predict enhancers that are going to be good, but there's lots of factors that could make it fail in practice. So you have to kind of still screen these to find the ones that work. Bigger picture, we're also interested in kind of applying these studies to understand questions about the brain. So for example, we're interested in understanding inter-individual variability. So all of these data come from laboratory mice that are inbred. So they're all genetically identical to each other, but we know that human beings have genetic differences. And so we have a project where we're going to be collecting brain tissue from postmortem samples of human brains from 12 different individuals, and we're going to look at how similar or different are the actual cell type specific methylation patterns across those individuals, and also whether they're affected by age. So some of the individuals will be young, some are going to be old. So we're interested in how aging affects the pattern of epigenetic regulation. Yeah. Okay, Hong Yi, could you impute the gene expression across time instead of different measurements? That's a great question. So all of these data sets are snapshots, right? So we have to collect the brain tissue. We have to sacrifice the animal, collect the brain tissue, destroy the cell in order to collect its DNA or RNA. So it's not really possible with these kind of technologies to track the gene expression over time, even though we know that there's a lot of dynamics to the gene expression. There are circadian rhythms to gene expression. There are dynamics over time as a function of neuronal activity. So there is one technique there is a very clever technique called RNA velocity. Have any of you guys come across RNA velocity? So this was an idea for how to use single cell RNA sequencing data to try to infer something about not just what is the current pattern of gene expression, but also how is that pattern of gene expression changing over time? In what direction is the cell moving? So there's a little bit like that, but I think that's still a big open question, is how can we make these measurements on a kind of more time-resolved way? Another approach that we're pursuing is you just look in at brains at different ages. So if you're thinking about kind of dynamics over the lifespan, you can look at brains from mice that are newborn or 1 week old, 2 weeks old, etc. And that's something that we're pursuing with some collaborators as well. Yeah, so Crichton is asking about that developmental aspect. So exactly. So yeah, it's interesting actually. So I think I didn't show you. I think I didn't show you my slide about development. Let's see if I have it here somewhere. So it turns out that a lot of what I've shown you is based on a type of DNA methylation that's called non-CG methylation. So you guys may have heard of. You may be aware that DNA methylation tends to occur at CG positions in the genome. It turns out that neurons have non-CG methylation. They have methylation at CA positions. And that changes a lot during development. At birth, it's not there. It then accumulates over development. So that's something that we're very interested in looking at. And yeah, you can use this technique to try to track neuronal cell types over development. Well, maybe we should cut it off there. I know you guys have another meeting to go to.
Yeah. Thank you so much, Professor Mukamel.
Sure, my pleasure.
So for everyone else, the GBMs right now, I'll drop the Zoom link in the chat. So if you guys have time, please come out.
Okay, guys, take care. Stay safe.